Hello, everyone. Welcome to Introduction to Sequencing Data Analysis. This class is part of the UMass Biocore Bootcamp. And the goal of this entire course is to make you familiar with genome-wide sequencing approaches and the computational analysis that are essential to them. Some of you may have an immediate application in mind or already have collected a particular type of data you want to analyze. So you can think of the possible new ways to interpret it during this class. And for those who do not have an immediate application in mind, you can get started in genomics and plan your collection and analysis, and maybe think about how to explore the massive amount of data that already is available. So before we get to the technical part, I want to briefly motivate everyone and give you a brief overview of the power of these approaches to survey the transcriptional and the chromatin landscape of a cellular tissue. So in this picture, I'm showing you a common visualization for genomic data, which are coverage profiles in a genome browser. And this is spanning over 300 kb of the mouse genome, as you can see. The PRDM1 gene is represented on the top left. And below the gene, shown in black, are two different tracks for RNA-seq. You can notice that the peaks for these tracks correspond to the exonic regions of the gene, expected from the higher contribution of mature or spliced mRNA molecules to the libraries that were sequenced. So notice as well that there is not much signal for RNA-seq outside of the gene locus. And we can also generate libraries to survey chromatin accessibility, shown in blue from ATAC-seq, or histone modifications using ChIP-seq, such as for H3K27 acetylation in light green and H3K4ME3 in dark green. And these data allow us to detect regions that likely function as regulatory elements, including promoters and enhancers.
So here I'm showing you an example of an H3K4ME3 ChIP-seq library and an H3K27 acetylation library. And these are all useful, both the ATAC-seq and the ChIP-seq libraries, so that we can identify potential regulatory elements, which are shown here as these bars. We've identified them through the coverage data, and we can use it to try to understand the regulatory landscape of a given cell. So with this picture, I also wanted to point out that there is a lot of power in these studies when done in combination. So integrating all of this information can help us understand more than we would if we examined them separately. And to introduce one final dimension, we can also perform these studies through a time course. So you can follow cellular processes as they happen if you collect multiple time points. All right, so the way we like to think about this type of data is as a readout of a cell state. So cells respond to changes in their environment, for example, to a threat of a pathogen in the case of innate immune cells. And we can observe these changes in their transcription output or in their chromatin landscape. So one of the favorite systems we use in the Garber lab, because of how dramatic the difference is between a stimulated cell and an unstimulated cell, is this system of dendritic cells, which are stimulated with LPS, a molecule from the membrane of gram-negative bacteria. And through this response, the dendritic cells become activated and begin an entirely new transcriptional program. So the way that cells do this is by turning on a signaling cascade. So LPS will stimulate the Toll-like receptor 4, and this will eventually lead to the activation of transcription of several thousand genes involved in the cellular response for this pathogen, including cytokines and other types of immune genes. So the result of this change is that very dramatic, like I said. The transcriptional landscape of these cells changes drastically, and they upregulate around 2,000 genes and downregulate an equally large number of genes. And I'm showing you only part of them here in this heat map. And the heat map, if you're not familiar, is a way that we use to display the information of expression for a large number of genes in multiple conditions and replicates. And here I'm showing you the expression of the genes in dendritic cells from 2 human donors in multiple time points of before or after stimulation of these cells with LPS. And the red color you see here for these genes marks, oh, when I go there, it doesn't let me. The red color you see here marks high expression of these genes. And the blue color marks downregulation of these genes in a given time point. So you can see that many of the genes in this heat map show similar patterns of expression. And the reason that it's so easy to visualize this in this heat map is that we actually clustered the gene expression by the genes that had the same response across time. So they're organized in this way, which you will learn how to do through Dolphin. And it makes it easier for us to pick out on these specific expression patterns and responses. So we can, for example, focus on a particular cluster here that shows a very fast response to the LPS activation. So these genes all get maximum induction only 1 hour after the stimulation. And 1 hour is the time point we measured. So actually a lot of them, their expression level goes higher a lot sooner. And if we do a gene ontology enrichment analysis for the genes in this cluster, then we can detect if there are specific cellular processes that are overrepresented by these genes. So if there is any particular enrichment, and as you can see here, there's enrichment for several processes that make a lot of sense for the immediate early genes. So you can see signaling, cell communication, a lot of transcription by RNA polymerase, which makes sense for cells that are responding to such such a dramatic transcriptional change. All right, so another thing that we can profile, like I said, for these same cells is the changes that are happening to their chromatin landscape. So we can detect them using assays such as ATAC-seq, which will measure the increased accessibility of the genome, or we can measure modifications to the histones, such as a higher level of acetylation, which we can do by ChIP-seq, like I said before. And you can see that for a particular enhancer that I'm showing you here, what you see with the ATAC-seq is that throughout time after the stimulation, there's no change to the accessibility. This locus was already accessible to begin with. But there is a dramatic increase in the acetylation level of the histones in this chromatin area. And this makes sense because the acetylation of lysine 27 in histone 3 is a mark that actually corresponds to higher transcriptional activity. So we use that to study the enhancers that are controlling genes that are responding to a particular transcriptional stimulus. All right, other ways that you can use these genomic assays for, other reasons you can use these assays for, are to, for example, examine changes that happen during differentiation. So here I'm showing you the hematopoietic cell lineage, and there's this beautiful study by Christina Leslie's group which showed all the regulatory elements that are common to all the cells, including the main stem cell progenitors, or common to only the hematopoietic progenitors and the hematopoietic lineage, or even specific to monocytes, for example, or any other mature cell type. And this helps you track the regulatory elements and the genes that are responsible for all of these transitions. And they did this by doing an assay called DNA-seq, which is very similar to ATAC-seq in terms of what the outcome will be. So the outcome will be, you will assay the areas of the genome that have higher accessibility. And one final example is that you can use all these assays, for example, for creating gene expression profiles in disease. So here I'm showing you another example of this study from a couple of years ago, where they compared neuronal stem cells and mature neurons from both autistic and control individuals derived from fibroblasts. And they could see several genes that were dramatically changed in their transcription and make successful conclusions about what kind of processes were changing in this disease and how this was reflected in the transcriptome. So several of the phenotypes that they observed for these neurons that made sense given the patient alterations, you could see a reflection of that in the transcriptome and find this particular pathway that was leading to that phenotype. Okay, so the thing is, how do we get there? I've shown you all these nice paper-ready figures and conclusions from these studies, but a lot of you may be wondering, what are the steps that I actually need to get there? Okay, so this is what we're going to go through, hopefully for the whole rest of the course, and by the end of it, you'll be able to get to these figures and analysis with your own data. All right, so let's go through a quick overview of what needs to happen. Let's say you have some cells of interest and you generate a library of fragments of cDNA or DNA from these cells, and you submit that to a sequencing machine. All of this is happening in the wet lab, and after this, what you get is a set of reads. So these reads are contained in files in your computer now, and we need to figure out, first of all, where in the genome they came from. So mapping is one of the biggest challenges we have, and is the first thing that you're going to do with your reads before you can interpret the results. And after we know where they came from, then we can go through these analyses where we generate read coverages and interpret the results that you obtained. So we will go through the computational steps required for this in more detail. But this is the basis for, and we're going to go through in detail for RNA-seq, but most of these steps are very common between RNA-seq and any kind of genomic approach, such as ChIP-seq or ATAC-seq. So it can be useful for both cases. It's just that we're going to focus on a particular pipeline for RNA-seq. And with that, I'll finish this first part, and you guys can go through the genomic resources section. So let me exit here and show you again. Just go to the genomics resources link now and follow this, where you'll learn how to find the reference genome and what to expect in the genome folders that we have in the Biocore. And if you have any questions, I'll be happy to answer also while you go through this.
So how about 10 minutes for this?
Yep, 10 minutes is good.
Okay, so we will continue on 10:45. Please finish this. We will keep answering your questions. So let us know if you have any problems anywhere. Okay, you can start now. Thanks, Elisa.
You're welcome.